So this second session of the day, we're going to focus on vaccination and immunity. So we're diving down into within host modeling, looking at what's going on within people and looking, I guess the goal is to look at what's driving the immune landscape that this pandemic to endemic transition will hopefully happen smoothly on. So our first, we have three speakers and then a panel discussion. So we have Jane Heffernan and Alison McGeer and Morgan Craig, and then hopefully Syed Mohagas will join us for the panel discussion. Jane is a professor at York University, lots of experience modeling both within and between host infection, both at the same time too. She leads our national team on in-host modeling, sits on the Canadian Immunity Task Force, and has a York Research Chair in multi-scale methods and evidence-based health policy. And so I'll let Jane take it away.
Thank you. I'll just share my screen. And so what I thought about talking about today is, well, modeling immunity, but to really just give a sense of why we're interested in modeling immunity. And so just as a reminder, so immunity is something that we think about in terms of an individual acquiring immunity, say from infection or from vaccination. But a goal of modeling and of public health also is to get a sense of what the distribution of immunity is in a population, so kind of like called herd immunity, so that we can get an idea of what the overall level of immunity is in a population to see if we can protect that population, or to see if we don't have enough immunity, then we implement vaccination booster programs, or maybe we need to put in some public health mitigation strategies so that we can lessen the burden of infection. And so when you think about immunity at the individual level and the population level, you have to think about how they're coupled to these two levels of immunity. And so we have got transmissibility. So an individual can transmit an infection or transmit a pathogen to other people in a population. And their transmissibility depends on their viral load, and the viral load in their body, or the pathogen load in their body, depends on more or less what their immunity looked like at the time that they were infected. And so then we need to think about that in terms of susceptibility as well. So when we're an individual circulating in the population, we have to think about when we're coming into contact with other people and there's an infectious disease being transmitted in the population, how susceptible are we to that infectious disease? And our susceptibility is going to be determined by all the different infections that we've had in our history that are related to the pathogen in circulation, but also all the different vaccinations that we've had over our life history that are also related to that pathogen. And so when we think about modeling immunity, we need to think about this feedback. And so we need to think about modeling how your immune system interacts with the pathogen and how immunity is developed. We need to look at how maybe that immunity can also wane over time or decay. And so what's the effect of that immunity if you're exposed to a pathogen a couple of weeks after you're infected or a couple of weeks after you're vaccinated versus months later or years later. We can also use in-host modeling to give us an idea of how severe someone's symptoms might be. We, of course, can look at how immunity is generated from different types of vaccines for different pathogens as well. And then when we're thinking about all of these different aspects of immunity in an individual, we then need to take into consideration the behavior of an individual as well, because that's going to also affect their susceptibility and also affect their decision-making and how on whether they're going to go and get vaccinated. So we incorporate those components into our models too, when we're thinking about population level immunity. And then of course, we know that the distribution of immunity is different all over the world in different populations against different pathogens. And so when we're in a particular city like Toronto, we can consider all the different immigration and emigration rates from other countries in the world as well. And we can see what the immune systems look like in our immigrant population and in our population that's, say, born in Toronto, so that we can get an idea of what the distribution of immunity looks like in Toronto and how it differs from, say, the distribution of immunity in Saskatoon. Okay. So ultimately, when we talk about immunity at a population level, we're interested in herd immunity, but this really is related to seroprevalence. And so we're interested in understanding what the distribution of immunity is in a population, and that's called seroprevalence, and we can measure that. So we can measure antibodies. But underneath that level of seroprevalence, there are also different components of the immune response that also provide immunity to individuals. And so I'm not going to go into a lot of detail about the immune response in this talk, and I'll leave some of the discussion about the immune response to Allison later. But more what we want to do is we want to have a sense of how we can measure immunity in individuals and how that's translated to a measure like seroprevalence in a population, which can then help us determine whether a population is well protected or whether we need to implement more vaccination program or whether we need to bring in public health mitigation strategies to dampen the severity of disease. Okay, and of course, that's also going to be affected by seroreversion, which is also called waning immunity. So we have people who seroconvert and we have seroreversion, so our decay in antibodies over time. And then we need to think about as well all these other components of the immune response that help us have immunity against infection or against severe disease. Okay, so here is a schematic of a mathematical model for an in-host model for influenza. And I really just wanted to show you what this looks like and how complex it can be. And so we have all these different types of immune response components. So we have T cells, Th1 and Th2. We have B cells, we have antigen presenting cells and pathogen. And you can see a list of what all of these are on the right-hand side. So when we're looking at in-host modeling so that we can look at the development of immunity and the activation of the immune response against a pathogen or activation induced by vaccination, we want to keep track of important components of the immune response that are going to allow us to determine what immunity might look like in this individual post-infection or post-vaccination. But this is a very complex model, and we know that when we're modeling, the larger the model gets, the more complex it gets to analyze and the more uncertainty we have. And so what we want to do is we want to simplify these models down to a few specific components that we can measure and get some data from, or some data to inform, so that we can have more confidence in our model output. And so when I talk about immunity in in-host modeling, mainly what we focus on is outputs like antibodies, and this is related to B cell memory. We also look at T cell memory like CD4 T cells and CD8 T cells. But then there are other components of the immune system that can be measured that give us an indicator of what these populations, what these other populations like B cells and T cells look like. Okay, and then when we're developing our in-host models, we always need to think about this optimization program here, where what we want to do is we want to incorporate enough biology into our model such that we get an optimal level of new knowledge. So if we incorporate too much biology, we're going to put in a lot of uncertainty and we're not going to have a lot of new knowledge from our model. And if we don't put in enough biology, then we can't actually answer the question that we are tackling, and we'll have no new knowledge as well. We want to live up here at this peak. And of course, as laboratory technologies get better and get cheaper, it'll be easier for different measurements of the immune system to be done. And so you can think that this peak will move move to the right as that happens. Okay, so I just wanted to give you an idea of some of the in-host modeling projects that people in Canada have been working on during the pandemic. So this is a model of an adenovirus vaccine and how it incites activation of the immune response in an individual. So here we have cells like T helper cells. We measure things like interferon gamma and interleukin-6 that help us get an indicator of what plasma cell activity is in the body, and plasma cells are related to antibodies. And then effector cells can go and kill infected cells in the body as well. So what we want to do is we want to get an idea of what the immune response looks like. And we have taken this larger model that you can see in the background, and we have simplified it down to just specific components of the immune response that we have some data for that we can use to model immune system activation and memory development. And so what you have on the right-hand side here is so in the data, we have an adenovirus vaccine data for 2 doses and 1 dose for different age groups. We fit this model on the left to that data for 2 doses. And we try to see what happens when we have 1 dose, which is the blue line, and 2 doses, which is the red line over time. So we're looking at antibody concentration here. And then we do the same thing while we're fitting to antibody data, we're also fitting to interferon gamma data here. And so you can see that if we have 1 dose, we have this increase in interferon, which is related to our effector cells, and then a decrease. And then we have this, with the 2nd dose, we have another increase and a decrease. So after we fit this model to our clinical data, then what we've done is we've tried to see, well, what happens with our vaccine model when we have, instead of having 2 standard doses that are separated 28 days apart, what if we have more days between the 2 doses, so a longer period of time? And so we can look at our antibody output and our cytotoxic T cells as well. Or what happens if instead of 2 standard doses, we look at a standard dose and then a half of the dose for our second dose, or a quarter dose, and so on. And the idea is to use mathematical modeling to try to determine what the antibody output and the cytotoxic T cell output from these different scenarios will be, so that if we need to somehow mitigate or lessen, or if we have a restriction on the amount of vaccine that we have available to a population, mathematical modeling might be able to say, well, maybe this will happen, and then we can try to test for these things in small clinical trials or in the lab. Okay, so here is another model that we did. So we took a very similar model to what I just showed you, and we fit it to data from clinical trials from Moderna, which is here in green. So this is the mRNA 1273, and Pfizer, which is BNT162b2 here. And then the blue is a combination of the 2 in a separate study. And so we took the same model, we fit it to this data, and then we looked at what the decay in our antibodies would be over time to see if we were going to look at a booster dose in the from peak value. And so by day 190, we have 99% loss of our antibodies. For Moderna on its own, by day 238, we have 99% loss. And so while we were doing this work, we were considering, well, when would a booster dose be needed in a population? And this is irrespective of age. And so we considered that by day 200, probably a 3rd dose of vaccine could be suggested for the population. And at the same time that we were doing this research, the CDC in the US came out and they modified what their recommendation is for the 3rd dose of vaccine, and it's in line with what we were seeing mathematically here. Okay, we can do the same for looking at males and females and looking at a different immune response from the two vaccines and what the decay would look like, as well as by age. And so what we want to do is we want to take all this information from vaccine studies and also studies of other things, like what our immune system outcomes look like from infection, mixing and matching of different vaccines, infection then vaccination, or vaccination and then infection, and see how what immunity looks like in all of these different individuals, and then put all of these individuals into a population so that we can get an idea of what the distribution of immunity looks like. Of course, then we need to also consider different variants of concern when they might arise in a population. So we have models for that. We also have models on how these different variants can be transmitted between individuals and what the probability is of transmission. And then we can also look at immune escape mechanisms, like what we know for Omicron and how it's able to escape some of the vaccine-induced immunity, and what it exactly is doing, and how booster doses of vaccine might be able to mitigate that effect. So we take all this information about the immune response, and we can then incorporate that into a population level of model of immunity. And so the idea then is that we take all this information, incorporate it into a population level of immunity that we can then use to look at distributions or project what the distribution of immunity might look like in a population. And so here is a schematic of a diagram where we have people who are susceptible, totally susceptible in S1. And if they're exposed to the pathogen, then they can either get a mild disease, moderate disease, or severe disease. And the probabilities of mild, moderate, and severe infections for SARS-CoV-2, we assume, are dependent upon the prevalence of comorbidities in the population. And so we can split this model up for different age groups, and then we can look at what comorbidity status looks like for 0 to 4-year-olds, 5 to 10-year-olds, or 5 to 9-year-olds, and so on, up the population. And then these probabilities of mild, moderate, and severe infection will change. We can also assume that after you have a mild infection, you'll gain some immunity, but after a moderate infection, you gain even more. And after a severe infection, you'll gain even more. But then we allow this immunity to decay over time, so waning and seroreversion, so that we can look at what different protective capacities are of the immune system after mild, moderate, and severe infections. We can also incorporate the vaccine into this type of model, where you can have 1 dose of vaccine, as that's at this level, or 2 doses of vaccine. And then the different shades of purple, we can modify the characteristics such that maybe the darker shade of purple means that you're totally protected from infection. But this shade of purple here could be that you have some high protection against infection, but if you are infected, you have a very high protection against severe disease. So you have a higher probability of having mild infections. And this other level here is that you have even a lower level protection against infection. And if you are infected, you'll still have protection against severe disease, but it will be less as well. So we can incorporate all of this information from our in-host models into population level models to then get an idea of what infections look like over all of these ages, over the entire pandemic, how vaccination uptake in all of the age groups mitigates the infection effect in all of these age groups, and then see what the distribution of immunity looks like over time. So then, of course, we have to consider what all of these things look like in the data. So we would assume that severe infections would certainly be reported. Moderate infections would have some probability of being reported, and that mild infections, which would be like asymptomatic infections, would only be reported if they happen to have a test, say if they were going to visit their grandmother, for example, or if they were contact traced. So then we can incorporate different scenarios for vaccination, the rollout by age, how public health mitigation strategies affect all the different age groups, like closure of schools or closure of work or closing the shopping mall or who goes shopping, all of that information into the model to get distributions of immunity. So we pull from the literature different components, And then we fit our model to data. So this is assuming different waning rates of immunity over time for different types of vaccines. We have a vaccine that provides a lot of neutralizing immunity and a vaccine that doesn't. So we fit this to data. So we have severe infection, moderate and severe, and mild, moderate, and severe for incidence. And then we can project what our distribution of immunity will look like over time. And so here we have in blue, we have individuals that are vaccinated with 1 dose. In green, individuals that have 2 doses of vaccine. And in red, we have all the different individuals that have gained immunity from infection. And so, or have had their immunity wane from vaccination. And so you can see what the burden of infection is pre the vaccination era. And then as vaccines increase in uptake in the population, then you can see the lessening of infection in the population, but you can also see where the burden, the greatest burden of infection is in the population, which is in earlier on, the older age groups, and then later on, we see a lot of mild infections happening in the younger age groups. Okay, so given that information, then we can really look at, okay, what public health mitigation strategies do we need to implement in the population for us to increase our immunity in the population to mitigate the pathogen? And so this is some work that we did back in August, where we were looking at what Ontario might need to look at in order for the fall wave that everybody was thinking was coming. And so here, using this mathematical model, we were able to show that individuals that are aged 12 to 29, that there wasn't enough uptake of vaccine in those age groups. So vaccination programming for the 12 to 29-year-olds should be incorporated, or more advertisement or some public health programming for that should be increased. But we also found that most of the infections that would happen in the fall wave would be mild, which is here in blue, or moderate in yellow, and not a lot of severe infections were going to happen. Okay, but on the right-hand side, if we were looking at our severe infections, then we're able to also see, because our model keeps track of who's getting infected, where the severe infections would be. And so we certainly see that if we're looking at 50% of the severe infections would be in the age groups 50 plus. And so we recommended that, that booster doses be rolled out to age groups 50 plus, and certainly for people who are immunocompromised in those age groups. And we recommended that early on for the fall wave. Of course, when Omicron came, we would have to extend the model to look at more distributions of immunity and how these wane. And so this gives you a little bit of an idea of how we're thinking about how waning immunity would be increased with respect to effective immunity for Omicron. And so you can see this decrease here, these decreases. That is in part due to an effect of Omicron coming in, but also a natural decay of immunity in the population. And so we're in the process of doing something similar to the previous analysis of looking at, I know this is kind of late, Omicron's already here, but trying to see how we can shift the model to give us a better idea of how these distributions of immunity are related to the immune escape mechanisms that Omicron has in the population in individuals. Okay, of course, when we're talking about immunity in a population and shifting people to have higher probabilities of mild infection, we also then need to think about what the ascertainment rate is going to be. And so if we think about testing rates similar to pre-Omicron, we found that our ascertainment rate from fitting our model would change from about 1 in 3 to about, I can't see my axis, to about 1 in 3 to about 1 in 14. So for every 1 infected case reported, there would be 14 cases, but they would all be very mild. Okay, but with the change in testing, especially in Ontario, with who's eligible to get tested, we would assume then that this ascertainment rate will be a lot different. And so we're in the process of modifying these models that incorporate immunity to look at what the possible ascertainment rate would be. So this work is done with a lot of collaborators, and it's supported by a lot of different organizations. And I think that I have used up my time, and I will turn now the floor back to James.